So, as many of you may know, the midterms are coming up for biology reasons, so I figured might as well make a video. I already have quite a few of the notes down already in other videos, but today I was thinking we could do the question and answers for the first 2 units, and I'm going to keep going until we got all the notes covered. So, Unit A and Unit B, ready to go. I'm going to say the question, you can think of the answer. Woo! Okay, I'm sick if you're wondering. Okay, so, Unit A. What are the steps of a scientific method? 1. Observation, which inspires the experiment. 2. Hypothesis. 3. Controlled experiment. 4. Recording and analyzing the results. 5. Conclusion. And 6. The theory. Second question. What is a controlled experiment? Controlled experiment. What is the definition? So, it's an experiment that has one group under conditions that will generate a known outcome, and a second group that has one variable that will result in an unknown outcome. So, 2 variables you got. So, what is the manipulated or the independent variable? The variable being tested or deliberately changed. It is shown on the x-axis, so independent x-axis. What is the responding or dependent variable? It's the variable on the y-axis that is observed and can change. Changes based off the experiment. So what are 3 ways to make an experiment more reliable and valid? So having a large sample size, being able to repeat it, that's necessary, and it has a clear procedure provided to prevent errors. So what happens in the conclusion of an experiment? Data is gathered and it's used to evaluate the hypothesis, which is made in the beginning. The hypothesis may be proven or disproved. When is a theory formed? After many hypotheses have been tested and proven true. What are the 8 characteristics all living things have in common? Cellular organization, reproduction, heredity, growth and development, metabolism, response to environment, homeostasis, and evolution. What is the characteristic of cellular organization? Just describe it. What is it? So, living things are composed of cells. Cells are the basic unit of life. Okay, so describe reproduction. Living things possess the ability to reproduce. This can be accomplished sexually or asexually. We've learned both so far. So, describe heredity. Living things possess DNA that contains the information necessary to pass on the ability to control cellular activity to their offspring. Describe growth and development. Single cells grow in size, but multicellular organisms go through a series of changes called development. Describe metabolism. Living things obtain and use materials and energy in order to carry out cellular processes. Describe response to environment. Living things detect and respond to stimuli. Describe homeostasis. All living things have the ability to detect and respond to stimuli. Describe evolution. A group of organisms can change over time. Name the levels of organization in biology from smallest to largest. Levels of organization. So molecules, cells, tissues, organs, organ systems, organisms, population, community, ecosystem, and biosphere. Okay, so what is length measured in? We're going with the decimal system. Meters. So what is volume measured in? Volume. Liters. What is the mass measured in? Grams. The decimal system is based on multiples of 10. Deci equals 0.1. Centi equals 0.01. Milli equals Milli equals 0.001, 2 zeros. Kilo equals 1,000. To sum it up, say them all in order, from largest to smallest. Oh yeah, you should just pause the video because you can't really answer that quick. Okay, so kilo, deci, centi, milli. What are the two main types of microscopes? So there's light microscopes and electron microscopes. So what is the difference between a monocular and a compound light microscope? So monocular and compound light microscope. So a monocular microscope has one eyepiece, and a compound microscope has more than one lens. What's the difference between a binocular microscope and a dissecting microscope? So a binocular microscope has two eyepieces, and dissecting microscope is 3D with lots of room between the stage and the objective lens. Okay, so what is the difference between a transmission electron microscope and a scanning electron microscope? So a transmission electron microscope lets you see inside of stuff, and a scanning electron microscope lets you see the outside of stuff. So inside transmission, outside is scanning. So transition versus scanning. Okay, so name 3 laboratory techniques. So there's cell culture / cloning, centrifugation / cell fractionation / cell separation, and there's safety issues. So that's kind of basic stuff. Okay, so now we're on to Unit B. Woo, that's kind of the longer one by far. But okay, so what is an atom? It's the basic unit of matter. What are the parts of an atom? You should just know this from living. So there's protons, neutrons, and electrons. What's a molecular bond? So it's a bond that holds 2 atoms together or more. So what is needed to make a molecular bond? Energy. What is released when a molecular bond is broken? Energy. What are the molecular bonds also known as? Covalent bonds. What is polarity? So that's when some molecules share electrons unequally. These molecules have a slightly positive side and a slightly negative side. So that's polarity. So what are 2 examples of polar molecules? Water and sugar. What's one example of a non-polar molecule? Oil. Polar molecules will blank in polar substances. Dissolve. What is an example of an organic molecule? What is an organic molecule? So that's a molecule which contains both carbon and hydrogen atoms. So what is an inorganic molecule? It's a molecule that does not contain both carbon and hydrogen together. What is a molecular formula? What is it? So it's a formula which shows the types and number of atoms in a molecule. What is a structural formula? It's a diagram showing how the atoms are arranged in a molecule. What does a chemical reaction involve? Changes in the chemical bonds that join atoms in compounds. What does a molecular equation do? So it shows the molecules that were involved in a chemical reaction and what molecules were formed as a result of it. So what are the 2 parts of a molecular equation called? Reactants and products. So what does the arrow symbol symbolize? That symbolizes a chemical reaction. So what 4 elements make up over 99% of your body? Carbon, hydrogen, oxygen, and nitrogen. What are the 4 major groups of organic macromolecules found in living things? Carbon, carbohydrates, lipids, proteins, and nucleic acids. What's the function of carbohydrates? So they're the primary energy source, and they have some structural functions as well. So what are carbohydrates composed of? Carbon, hydrogen, and oxygen. What are the types of carbohydrates? Monosaccharides, disaccharides, and polysaccharides. What are monosaccharides? They're simple sugars. They're the monomers of carbs. What's the molecular formula of monosaccharides? C6H12O6. What are some examples of monosaccharides? glucose and fructose. What are some examples of disaccharides? Sucrose, which is table sugar, and lactose and maltose. How are polysaccharides formed? By joining many glucose molecules. What's the storage form of glucose in plants? Starch or glucose? What's the storage form of animal muscle cells and liver cells? Storage form of sugar in animal cells. And, glycerin. What's glucose found in cell walls that is indigestible by animals? Cellulose. What's the form of glucose found in the exoskeleton of insects? Chitin. What's the function of lipids? Long-term stored energy, insulation, protection, and the cell membrane. What's the composition of lipids? Carbon, hydrogen, and oxygen. What are the monomers of lipids? Glycerin and 3 fatty acids. What are the fats formed mostly by? Animals. What phase are fats in room temperature? So like liquid, solid, vapor. I don't know if that's what the word is. I forgot. Solid. Okay. So what do fats lead to? Cholesterol formation. What do fats contain? Salt water, fatty acids, saturated. What are oils mostly formed by? Plants. What are they at room temperature? Solid, liquid, so they're liquid. What don't they lead to? What don't oils lead to? Cholesterol, no cholesterol. What do oils contain? Unsaturated fatty acids. What are some other lipids, not fats or oils? So there's cholesterol, steroids, wax, and phospholipids. What is the function of nucleic acids? That was okay, anyway. So it stores and transmits genetic info, controls cell activities, and directs formation of proteins. What's the composition of nucleic acids? So carbon, hydrogen, oxygen, nitrogen, and phosphorus. What are the building blocks or monomers of nucleic acids? Nucleotides. What are the types of nucleic acids? Don't overthink this. So there's DNA and RNA. What is a function of proteins? So there's structure, cell membrane, and hair and fingernails, and connective tissue. And there's enzymes that aid chemical reactions. So what is the composition of proteins? Carbon, hydrogen, oxygen, nitrogen. What's the monomer of proteins? Amino acids. What happens if the shape of a protein changes? It stops working. What can change the shape of a protein? High temperatures, changes in pH level. What can slow down an enzyme? Low temperatures. What are enzymes? Organic catalysts and proteins that lower the activation energy and or change the rate of reaction. What is activation energy? The amount of energy required to start a chemical reaction. What is activation energy usually in the form of? Heat, which changes the shape of an enzyme, if you're thinking about it. Okay, so what is the structure of an enzyme? Changes the shape of the protein, sorry. What's the structure of an enzyme? It's large globular proteins. So the shape of enzymes, blank, closely corresponds to the shape of the blank. So the shape of an enzyme's active site closely resembles the shape of the substrate. So the substrate is that thing that goes into the protein. Enzyme. So how are enzymes named? The substrate name plus you replace the ending with -ose. So example would be maltose, maltase. So ace, I guess is what you replace it with. So what temperature do humans and mammal enzymes work best at? 38 degrees Celsius. What pH level do stomach enzymes work best at? 2. What pH do enzymes in the small intestine work best at? 8. So there's some variety. So you need to control enzyme activity to maintain what? Ooh, questions. Homeostasis. What is pH? It's the measure of acidity or basicity of a solution. How do you measure pH? So you test for the H+ ions of a solution. More H+ equals acid and less equals base. So I really hope that helped, and I'm going to be making more as we get through the unit. So, up a lot more. But thank you. I hope this really helps you study.